Hi, my name is Reenu and I'm a research scientist here at ALS TDI, and I'm going to talk about gene expression and how that fits into our organization's role in trying to find a cure for ALS. So gene expression is essentially what it is. It's the study of how genes are expressed, and because the mouse is our model, we mostly study gene expression in the mouse. So the way gene expression works is that there is the dogma of molecular biology, and what that illustrates is that DNA stores the information, RNA transcribes that information in the nucleus, and then that information that the DNA wants to pass along is then translated in the cytoplasm via proteins. So that's really what the dogma of molecular biology is, and where we fit in is the transcribed message. How is the message transcribed based on what kind of perturbations you're putting into a cell. So assuming you're giving a drug to a mouse, you want to know what is the DNA trying to do. What is it? How is it trying to overcome? As a result of injecting, say, a drug in a mouse. And as a result of that, what kind of message is it transcribing? So in a way, we're trying to capture that snapshot of the transcribed message. So the way we do that here is, say, we put a drug in our mouse, and then what will happen is our amazing lab technicians will then extract the tissue that will then get passed along to me, and what I will do is process the tissues so that I can extract the RNA. Remember, keep in mind that the RNA is the carrier of the transcribed message. So the RNA is then extracted on this. We have an automation machine here, and here at TDI we believe in having a lot of things done in high throughput, and the way we do that is we usually extract a lot of our samples in the 96 format. The reason we do that is because it just gives us more power to our studies, and in addition to that, it shortens the time, the processing time. If I were to process these 96 samples, it would probably take me 3 to 4 days. But on this machine, a couple of hours, 3 to 4 hours, and all 96 samples are processed. And then we'll go through a couple of iterations of prepping the RNA so that it's ready for the microarray. And we have a top of the line gene titan that helps us study gene expression. This machine is very cutting edge. One of the few that we have in the area, and what this encompasses is it has a scanner, it has a wash station, as well as a hive oven, and this is, I think, the first of its kind that's on the market. And what we do is we put we spot our prepped RNA cocktail onto these pegs that are on this 96 array. And essentially, each of these pegs have the mouse genome on them, and that mouse genome consists of about 39,000 transcripts that are measured, and we have about 45,000 probes that are, on these pegs to measure those transcripts. So once the data is, once it's been hived and washed and scanned, then the data will come out, and we have in-house bioinformaticians that will look at the data. These data are there are millions of data points that come off of this machine, and it will take about 2 days, to process the data set. And essentially, what we're trying to see is, assuming we did put a drug in, what did the drug do? Was it efficacious? If it was efficacious, how, then we go back and we see how long we treated our mice, and assuming, could we increase the efficacy by increasing the dosage, or maybe increasing the time regimen for giving the to the mice. So really, that's what we try to do. Whether we did get closer to a therapeutic goal or not, and that's what we are trying to do here. Thank you.